Good afternoon, everyone. Today, 26th of March, we are going to discuss some aspect in cell biology. As you know, my uh, emphasis always remain in the cell. So, starting with the cell, I have shown you a cell and animal cell with cell membrane. This is the nuclear membrane, and this is DNA molecule, and it will form chromosome and then centriole cytoplasm. And I have enlarged this cell this way to show you endoplasmic reticulum, Golgi apparatus, cis Golgi, medial Golgi, trans Golgi. So when you think about a cell, it always moves through a cell cycle, and the cell cycle means it tries to maintain its constancy in the body uh, through the process. So to maintain its constancy, it undergoes cell division. So I have drawn here a somatic cell. So somatic cell means your cell, human somatic cell. So any cell, liver cell, Kupffer cell, your blood cell, epithelial cell, skin cell, everything. So all is diploid and retained 2 into 23, 46 chromosome. But in the somatic cell also, the chromosome constitution bearing 44 autosomes, and then for the male XS and female XY. So as this diagram has shown you that there a single cell, a blue color DNA molecule is encircled by a nuclear membrane. It will divide and produce two new somatic cell. But thing is that special feature it follows that its size remains same, and all the cell cell organelle organelle remain constant. So this cell, before going into the division cycle, it maintain a phase interface, that is, it will increase its suppose this is the cell and here you have DNA molecule. So it will during the synthetic phase, this DNA molecule also become double, and after that this prophase, metaphase, anaphase and telophase will come. So it is better to understand a DNA molecule first, because every cell has a DNA molecule and two antiparallel sheet or antiparallel sugar phosphate backbone moves, and it is basically formed with some adenine, purine, that is adenine and guanine. So purine. Purine, as you can see, two uh, six co- six membered ring, carbon and nitrogen, and another five membered ring. Both has this amino group, and up to this is adenine, and then you have deoxyribose sugar. This is deoxyribose sugar as 2 prime deoxy, and then their phosphate group. From the phosphoric acid, this will attach. So, uh, and our cell membrane, it is of glycerol and fatty acid. So, this will form a primitive cell, but our cell has particular uh, purpose in our body. So, they produce, suppose it is a B cell, plasma cell, so it will produce a particular type of antibody. So antibody, the expression of antibody, that is the protein, is dependent on this B cell. So it is cell specific production, because you will not see any other cell will produce antibody. So this is another point, that DNA within particular type of cell express differently. So now my purpose to discuss this thing, that suppose from a particular cell, if you understand this, that is already I have discussed several times, that your DNA molecule, while run in a two opposite direction, 5 prime to 3 prime or 3 prime to 5 prime, you will see that some adenine binding or remain hydrogen bonded with thymine. So here I have also shown the very clearly that carbon and its alternating nitrogen, and its role to form a hydrogen bond with thymine. Separately, same if corresponding anti-parallel sheet from a template is forming, then the subsequent nucleotide is added, and it actually codes for the corresponding one. So if it is C, so it will be G guanine. So likewise, as I have shown here, you see G had same nitrogen and carbon in the ring. The position is shown here, and they will form a bond with C, cytosine. These are pyrimidine, cytosine, and they will form 3 hydrogen bond. So these subsequent nucleotide when come, they will bind this way also, because here we will have another this phosphate group. So this phosphate will bind CH2OH, so it will bind with this corresponding so it will form a bond with a phosphodiester bond with the incoming nucleotide, the same strand. So in this way you will find some helix is forming, and this way the cell generates 4 strands. Suppose you can see here, the previous strand will form another corresponding to the red nucleotides as a template. So now the question arises, what happens when a fertilization takes place? Because we know that our body cells bearing one chromosome from our mother and one chromosome from our father. So it in a functional state, in a homologous pair, it expresses differentially in various organs, various location of our body as a diploid state. So the protein we get from the, that means whether it is in a dominant position in the locus, it gives some express homozygous condition, heterozygous condition. So we get. But when we send our chromosome by the process of meiosis, you can see here your spermatogonium cell. So you will get ultimately the sperm. So this sperm will bear, or before producing this, it will have certain meiosis. So now you can see their body cell, when normally divides, it is mitosis. But when you when body perform meiosis, then meiosis, then actually this process takes place. So during this process, meiosis, because some part of the meiosis happens after fertilization. So meiosis is a process where recombination takes place. So the previous set of gene now get a scope to reassort, to substitute its place from one to another, recombination. So in this way, recombination when takes place in the sperm within our body, seminiferous tubule, where sperm cell undergo meiosis, our parental fertilized state, and gradually we form a present day organism, but that constitute, we get what we get as a gene from our parents in the somatic cell. They also get some chance to reassort themselves. Recombination takes place. So now when these from the both sides, female and male, and when they get fertilized inside the human newly female, yours, the sperm now, the recombined sperm from both the line, the mother line and the father line, they within their ovary and seminiferous tubule, they have been assorted, recombined, and then they are fertilized. So here the gene within the locus become very much, there is a huge chance of new locus arrangement. This is the locus. So suppose a globin chain of anemia. So if the parents, one of the parents from their father and mother has some defective piece, and the mother suppose had a normal beta hemoglobin. So these genes, when they particularly in a baby or inside a newborn, they have, after recombination within the respective mother and father's germinal organ, they will a new, that means constitution of the locus within a human baby body system. So it's a quite complicated thing, because if the genes are assorted in a sperm in a defective way, so it depends on the baby's chromosome setup whether or two both the genes are defective from the mother and father, or either one is defective. So if both the genes copies are defective in a homozygous state, so the baby will suffer severely, severe anemia and subsequently follow death. So this all depends on our gene chromosome. Basically, it's a understanding. So here all things very much dependent on this gene and chromosome. So nowadays everyone is working on this topic, how the cells, how you can isolate the proteins. So it's a very tedious work, because isolating particular protein is a difficult task, because when you uh, isolate the protein from a particular cell, there are other proteins also. So let us check in our next video how can we discuss this topic in a more constructive way. This is another thing, DNA and RNA. I have shown the 2' deoxyribose C2, where hydroxyl group is not present, only H is present. So thank you. Hope you will enjoy this topic.